Up until now, we've been talking a lot about chemistry. So we started off the quarter by taking a look at a review of chemistry. Specifically, we looked at the kinds of bonds that can hold atoms together and the kinds of attractions that can hold molecules together. Then just recently, in the latest knowledge check assignment, you took a look at the big molecules that you find inside of cells. So you took a look at what those molecules are, how to tell the difference between the major molecules inside of cells, and what each one of those molecules does. So you might be thinking, well, the name of this course is cell biology. What's the deal with all of this chemistry that we're taking a look at, and how does chemistry relate to cell biology? Well, so for most of the rest of the quarter, what we're going to take a look at is the way that cells work, basically. And so in order to understand how a cell works, it helps to know how the molecules that make up a cell work, and so what are the properties of those molecules that make up the cell. So that's why we're doing all of this chemistry stuff. What we're going to do in this video is build on some of the concepts that you learned in the knowledge check assignment on the molecules of cells. So specifically, what we'll do in this video is take a look at these two things. We'll take a peek at some of the shapes that are formed by nucleic acids. And so if you want to use the technical terms, we're going to take a look at the secondary shapes and the tertiary shapes that are formed by nucleic acids, the secondary and tertiary structures formed by nucleic acids. And then later on in the video, we'll take a look at what might cause lipids to dissolve. So that's kind of an interesting story, and it's something that is very relevant to cell biology. So before we jump in and take a look at these concepts, we'll first do a quick review of what you should have already learned in that concept or that knowledge check assignment on the molecules of cells. And so if you have been studying effectively, and if you've been understanding everything up to this point, you should be able to identify what these molecules are. So all of these things here are, again, major molecules that you find inside of all cells, and you should be able to look at their structure and at least identify what they are. If not, we'll go over them again, but if you're not able to identify what these structures are at this point, I'd really suggest going back and looking at that, because this has been kind of a major thing that we've discussed so far. So first, if we start with this guy right here, hopefully you recognize this as a molecule called an amino acid. And if you didn't recognize it as an amino acid, there's actually a way that you might be able to sound it out based off of other things that you know. So if you consider this portion of this molecule, this portion on the far left, you see that there is a nitrogen atom bonded to some hydrogen atoms. And so you might remember that the name of that chemical group, it's called an amine group. If you consider this portion of the molecule over here that has a C double bond O and then a hydroxyl group, that's called a carboxylic acid. And so if you can at least recognize this group, or these two groups, this is an amine group and this is a carboxylic acid, that's the reason why this entire molecule is called an amino acid, right, based off of these two groups and what those two groups are called. Another thing that hopefully you know up to this point is what amino acids form if you string a whole bunch of them together. And so hopefully you remember that a bunch of amino acids that are bonded to one another, what that forms is something called a protein. Hopefully also you recognize what this molecule here is. It has a 5-carbon sugar, it has a phosphate group, and it has a nitrogenous base. So molecules that have those three components are called nucleotides, and hopefully you remember that when you attach a whole bunch of nucleotides together, the molecule that forms is something called a nucleic acid. Hopefully you recognize this molecule here as, well, specifically this is a glucose molecule, and glucose is an example of a monosaccharide, and monosaccharides are a type of carbohydrate. So that's what this guy is right here. And again, from the knowledge check assignment, we talked about how you might be able to recognize carbohydrates as molecules that have a chemical formula that is some multiple of CH2O. That's why they're called carbohydrates. They are hydrated carbons, right? Carbons that have an H2O group attached to it. And then lastly, hopefully you also recognize what this molecule here is. And so again, this is another molecule that is you find a lot of inside of cells. Probably the giveaway as far as identifying what this molecule is that it has a large hydrophobic component to it. So there's a long string of carbons that are attached to nothing but hydrogens. All of those electrons are being shared equally between the carbons and hydrogen, so there are no partial charges on this part of the molecule, which means that it's not able to bind to water. And so the type of molecule inside of cells that have that property those are lipids. And so specifically this is a type of a lipid called a fatty acid. So again, just a brief review of the 4 major molecules that you find inside of all of cells. In the knowledge check assignment on molecules of cells, we talked a little bit about proteins and the different shapes and the different structures that they can form. So you might remember that we said that, so if you have a string of amino acids, again that's what makes up proteins. You might remember that there are 20 naturally occurring amino acids, and what makes them all different from one another is a part of the amino acid called the R group. And so I'll let these lollipop-looking structures represent the R groups. You might remember that different R groups from different amino acids have different chemical properties. So for example, some amino acids might have an R group that has a partial negative charge on there. Some amino acids might have an R group that have no charge on it. Some amino acids might have R groups that have a full positive charge, and some amino acids might have an R group that have a full negative charge. Depending on the sequence of amino acids, the shape that the entire protein forms will depend on the chemical properties of these R groups. So for example, if you were to put this whole protein in a beaker of water, this protein would take on a shape that would be dictated by the chemical properties of these R groups. So for example, you might see this happening. If you were to put this protein in a beaker of water, you know that opposites attract, and so plus and minus are opposites. You also know that these R groups that have partial charges on them, those would bind to water molecules. In other words, they're hydrophilic. These R groups that have no partial charges on them, or no charges whatsoever, would have no way of binding to water molecules, so they would be hydrophobic. So if you were to put this thing in a beaker of water, something like this might happen, where the opposite charges attract. The parts of the molecule that are hydrophobic would be hidden away from the water, and the parts of the molecule that are hydrophilic, the ones that have a partial charge, would be out in contact with the beaker of water. Right, so that the chemical properties of the molecules that make up a protein can determine the way that it ends up folding. And for a biologist, studying the way that proteins fold is a big thing, because cells have many different proteins, and they all have different functions. And what allows a protein to have a particular function is its shape. So again, these are all proteins that you find inside of cells. They all do different things. The reason why they can do a particular function is all because of its shape. So again, this is all a review. The thing that we're going to add on now is the fact that it's not just proteins that can produce shapes, that can fold into different shapes. Turns out that nucleic acids can also form secondary structures and tertiary structures. And so we'll take a look at how that happens and what the consequences of that are right now. So again, the bottom line from this slide is that a protein's shape determines its function, and different proteins have different shapes. Alright, nucleic acids. They can form shapes too. And so these are the 2 main types of nucleic acids that you find inside of cells. Hopefully you recognize the one on the left as being a type of nucleic acid called DNA, and the one on the right here is a type of nucleic acid called RNA, or ribonucleic acid. Typically inside of cells, for the most part, DNA is going to be double-stranded, whereas RNA is going to be single-stranded. So that's one way of telling apart DNA from RNA. What both DNA and RNA have in common is the fact that they're both nucleic acids, right? So that's what the Na stands for. Na stands for nucleic acid. And all nucleic acids are made up of nucleotides that are covalently bonded to one another. And so hopefully you recognize where the nucleotides are in each one of these molecules. And if not, I'll highlight one of them for you. There you go. There's a nucleotide. And again, In RNA, it's just a single strand of these nucleotides strung together that makes it up. Whereas in DNA, it's two of them, two strands that have been bonded to one another. So there's one string of nucleotides, and there is another string of nucleotides, and then they both come together via hydrogen bonding to form a DNA molecule. So hopefully none of that was news, because we mentioned all of this in the knowledge check assignment previous to this. Here's a new bit of information that we're going to add right now. Again, it turns out that nucleic acids, kind of like proteins, can form shapes. They can form secondary and tertiary structures. When you're talking about DNA, it turns out that these hydrogen bonds that form between the two strands of nucleic acids, they can actually cause the DNA molecule to twist, kind of like this, to form a double helix. Double helix because you have two strands of nucleotides and they're both twisted, so you form this double helix, or this twisted structure. And so when we say that nucleic acids can form structures too, this is the specific structure that DNA can form. Again, it's something called a double helix. It turns out that RNA can form structures too, and so we'll mention that next. So we'll take the DNA structure that it can form, kind of minimize it here in the corner so that we can focus on RNA and the structures that it can form. Because RNA tends to be single-stranded, that kind of gives it a little bit more flexibility than the double-stranded DNA molecule. And because it's more flexible, that means that it can form a greater variety of shapes as compared to a DNA molecule. And because it can form a greater variety of shapes, that means that it can actually do more things than a DNA molecule. So here's what I'm talking about. The thing that can cause RNA to form shapes is basically the fact that it can fold in upon itself. And the reason why it might fold in upon itself is because there might be complementary nucleotides within that very same strand of DNA that'll form hydrogen bonds with one another. So here's what I'm talking about in pictures. So what if we simplify this drawing of an RNA molecule so that it's just a pink string? So this whole thing is a simplified version of this. It's just an RNA molecule that is single-stranded. Again, you know that RNA molecules are made up of nucleotides that are all strung together. We mentioned that there are 4 major types of nucleotides that you find in RNA. What makes them all different is the nitrogenous base that's attached to them. So say, for example, that on this portion of the RNA molecule, you have nucleotides CUG found here. And say that a little bit further down on the RNA molecule, you have a string of 3 nucleotides that happen to be CAG. G is complementary to C. That just means that they would form hydrogen bonds with one another if they happen to be in close proximity with one another. U, you might remember, is complementary to A. C, again, is complementary to G. So if for some reason this RNA were to fold up on itself, kind of like this, because of maybe forces that are jostling it from the environment, there's a chance that this shape, that this structure would stick, because again, C is complementary to G, U is complementary to A, G is complementary to C. Hydrogen bonds would form between those nitrogenous bases. And so when that occurs, again, that makes this shape kind of stick. And so this is an example of a shape or a structure that RNA can form. It's an example of, specifically, it's an example of a secondary structure that RNA can form. And there's a term given to this specific structure that RNA molecules can form, and the term that's used to describe this might make a little bit of sense because it actually looks like this term. So this is a secondary structure that RNA can form that's called a hairpin loop, because this looks like a hairpin loop. Looks like a hairpin, something that girls might put in their hair to keep their hair in place, or guys might put in their hair too if they have long enough hair. One kind of cool thing to keep in mind is that RNA can be a pretty long molecule. It can be a pretty long string of nucleotides. And when you have a really long string of nucleotides, it's possible for a bunch of these hairpin loops to form within that same molecule. And so whenever that happens, you can get crazy looking stuff, kind of like this. Bam! Right, so what this represents, what this picture represents, this is all one single string of nucleotides. This is all one RNA molecule. But you have all of these hairpin loops that have formed inside of it. So you can get some really crazy shapes when it comes to RNA and all of the hairpin loops that it forms as a result of the hydrogen bonds that can form between nucleotides within that same molecule. And so because RNA can form all of these shapes, again, RNA has actually more functions as compared to DNA, which basically just forms one shape, which is that double helix. And so we'll sum that up here on this next screen. So if you compare and contrast DNA to RNA, again, one of the things that makes RNA different is the fact that it tends to be single-stranded, so it has more flexibility in terms of the variety of shapes that it can form. The consequence of that, so therefore, because it can form a greater variety of shapes, therefore it has a greater variety of functions, at least compared to DNA. And so here are the functions that RNA has. We already mentioned that RNA, probably the first thing you think of when it comes to RNA is the fact that it can store genetic information. So that's basically what nucleic acids do. That's one of their main functions, is that they store genetic information. DNA tends to be a more permanent holder of genetic information as compared to RNA, but they both have that function. They both store genetic information. Another thing that RNA can do is that it can act as a catalyst. So if you're not familiar with that term, catalysts, what they do is that they speed up chemical reactions. And if you're talking about something that speeds up chemical reactions inside of cells, those are called enzymes. So basically what we're saying is that RNA, because of the different shapes that it can form, some of those shapes might help speed up chemical reactions. So RNA has enzymatic activity. And so when people discovered this not too long ago, maybe 20, 30 years ago, it was very revolutionary, because at the time people thought that only proteins could act as enzymes. But RNA can act as enzymes as well, and we'll talk later on in the quarter that it's thought that RNA was the first molecule that served as an enzyme on this planet. Right? So kind of cool things. RNA can store genetic information. RNA can also act basically as an enzyme to speed up chemical reactions. Two really important functions that RNA has. If you contrast that with DNA, again, its major job is to store genetic information. Because it can't form all of these wacky shapes that RNA can, basically it's limited in what it can do as far as function. Basically that's all it does, is that it stores genetic information, but that's a really big job. So like I mentioned, DNA is kind of the permanent holder of genetic information inside of a cell. So don't feel sorry for DNA that it only has one job. It's one job is really, really important. Alright, so then that basically wraps up the one thing that we wanted to talk about in this video, which is that nucleic acids can form shapes. The other thing that we'll talk about in this video that has to do with the molecules of cells is lipid solubility. So we mentioned that water can't dissolve lipids. So if you put, say, some oil in a glass of water, that oil will stay oil. Those oil molecules will stay bonded to one another. Those water molecules won't be able to dissolve it. And the reason why that is because oil is a type of a lipid, and we mentioned that lipids have huge hydrophobic portions to them. So in pictures, again, this is a review of what we've already taken a look at. If you consider a triglyceride, and another term for triglyceride is a fat molecule, it has these long fatty acid tails that are made up of carbons and hydrogens. Carbons and hydrogens tend to share electrons equally, so that means that there are no partial charges anywhere within that part of the molecule. Because there are no charges in this part of the molecule, if you were to put a fat in a beaker of water, there is no way for these water molecules and their partial charges to grab a hold onto these fatty acids, right? Because there's no opposite charges to be attracted to. There are no charges at all on these fatty acids. So that's why lipids are not dissolvable in water. Remember that the kinds of things that are dissolvable in water are things that do have a charge. So for example, sodium chloride has full charges on it. It has a full positive charge on the sodium part and a full negative charge on the chlorine part. or the chloride part. So if you were to put sodium chloride or table salt in a beaker of water, those water molecules definitely have something to hold on to. There's some charge there to be attracted to, and if there's enough water molecules around, they could potentially split that sodium chloride apart, causing it to dissolve. So what we'll talk about next is, well, we've seen that lipids don't dissolve in water, but it turns out that there are things that can dissolve lipids. And so you might have heard this old adage that like dissolves like. And so that's, for the most part, true here. So water is a polar molecule. These fatty acid tails are nonpolar molecules. It turns out that if you want to dissolve a fat, which is a type of lipid, you have to put this in something else that is also nonpolar or non-charged. And so you might be wondering, well, how can a non-charged liquid end up dissolving a non-charged solid? So we'll explain that next. Why do nonpolar things dissolve other nonpolar things? So to be able to understand why that's the case, it'll first help to know something about partial charges. So we'll show you that here. We haven't talked about temporary partial charges in this class. You might have talked about temporary partial charges in chemistry, so we'll review that. So like you see up here, it turns out that even nonpolar molecules can have partial charges, but those partial charges are temporary. So let me back up here. So if you remember, we mentioned that by definition, if you're talking about a nonpolar molecule, it's a molecule whose atoms are sharing electrons with one another equally. So the consequence of that is that there are no partial charges that you end up finding, right? Because the electrons are distributed evenly across that molecule if it's a nonpolar one. What we're saying here, though, is that there are partial charges that can form on nonpolar molecules, but those partial charges that form don't last long. They're temporary. And it's because of these temporary partial charges that nonpolar things can form attractions to other nonpolar things. All right, so let's I'm going to explain why these temporary charges form right now. So say that this is a nonpolar molecule. So say that this is molecular hydrogen. So one hydrogen atom bonding with another hydrogen atom. The thing that bonds them together is the fact that they're sharing electrons. Hydrogen atom and hydrogen atom, they both have the same electronegativity. In other words, they are pulling on those electrons that they're sharing equally with one another. Here's the thing though, the electrons that they are sharing with one another may not be moving at the same speeds. Right? So let me show you that here. Because again, these are two hydrogen atoms that have the same electronegativity, this electron that they're sharing is going to spend as much time around the one hydrogen atom as it is around the other hydrogen atom. Right? So as a whole, this electron is spending as much of its time around this atom as it is around this atom. Same thing with this electron, right? So this is being shared equally between this hydrogen atom and this hydrogen atom. So you might see something like this if you could track what this electron is doing. Spending as much time around the one atom as it is around the other atom. But like I mentioned just briefly just a second ago, the speed at which these electrons are moving might differ, right? So this purple electron might be moving at a slightly slower speed than this green electron right here. Right? So one more time, that electron, that green one, is spending equal time around both hydrogen atoms as a whole, but it's moving relatively fast. This purple electron also is spending equal time around both of those atoms, but it's moving at a slower pace than that green electron. So there's a consequence to that. So because these two electrons might not be moving at the same speed, if you set both of them in motion and see what's happening, there are times when you'll see this happen. Right? So let's keep track of these two electrons as they're moving. One might be moving at a slightly different pace than the other. Because that's the case, you might end up with this happening, where one of those electrons actually catches up to the other one. And when that happens, check out what just occurred with this molecule. You have the electrons kind of bunched up on this right-hand side of the molecule, and you have no electrons over here on the left-hand side of the molecule. This is temporary though, so this charge that has just formed, so that there's now a partial charge on this right-hand side of the molecule, a partial negative charge. There's a partial positive charge on this left-hand side of the molecule, partial positive charge because it has lost its electrons. But those charges don't last long because these electrons are always moving. So a split second later, those electrons will become further spaced apart again. But again, because they move at different speeds, there are other points in time where you'll find this happening again, where the electrons are kind of evenly distributed so that there's one on this side of the molecule and one on this side of the molecule. So again, it's because of those different speeds at which electrons can move that can cause nonpolar molecules, even though they're sharing the electrons equally, sometimes they bunch up temporarily on one side of the molecule or the other in order to result in these temporary partial charges. So let's sum that all up. Right? So because these electrons that are being shared equally, even though they're being shared equally, they can temporarily bunch up on one side of the molecule. That results in the formation of this temporary partial negative charge on this one end of the molecule. And again, if the electrons happen to be bunched up over here, at least temporarily, this side of the molecule is going to have a partial positive charge. And again, it's also temporary because these electrons are going to end up dispersing evenly once again as they keep moving. It's the formation of these temporary partial charges that is kind of the key as far as why a nonpolar substance can dissolve another nonpolar substance. Right, so to write this down, we're saying again that even nonpolar molecules have partial charges. Those partial charges, though, are temporary if you're talking about a nonpolar molecule. The reason why this happens is because equally shared electrons can move at different speeds. And the result of that is that occasionally the electrons find themselves on the same end of the molecule, again resulting in these temporary partial charges. The consequence of that is again this. So say that you have two nonpolar molecules. Here's one nonpolar molecule. Here's another nonpolar molecule. We just mentioned that even nonpolar molecules can have partial charges, but they're temporary. So if this nonpolar molecule happens to meet this nonpolar molecule at just the right time, where this guy's electrons are bunched up over here, and this guy's electrons are bunched up over here, what ends up happening is this. There's this attraction that occurs between the two. Right? So this temporary partial positive end becomes attracted to this temporary partial negative end, and the same thing here. This temporary partial negative end becomes attracted to this temporary positive partial positive end. This attraction is temporary, right? Because these electrons are bunched up on these ends of the molecules only temporarily. And so there's a name given to this temporary attraction that can occur between nonpolar molecules, and the name of that attraction is a van der Waals interaction, or a London force. And again, it's the formation of these London forces, these van der Waals interactions, that's the key to understanding why nonpolar things can dissolve other nonpolar things. All right, so let's kind of redraw this in a different way. So again, if you didn't catch that, what a van der Waals interaction is, aka London force, is a temporary bond between two nonpolar molecules that can form because of all of this stuff here that we just explained. So if you keep that in mind, that nonpolar molecules can have partial charges that are just temporary, that can help explain lipid solubility. All right, so here's a huge nonpolar portion of a molecule. All of these fatty acids are actually nonpolar because they're made up of just C's and H's that share electrons equally. We mentioned that nonpolar molecules can form these things called van der Waals interactions, or London forces, with one another. That's what these green dotted lines represent. If you were to put this fat molecule in a beaker of a liquid that also happens to have nonpolar portions to them, when you're considering the molecules of that liquid, So say, for example, you put it in a beaker of ethanol. That's what each one of these represents. Notice that ethanol has a pretty decent sized nonpolar portion to it. So it has a pretty decent sized portion that is nothing but C's and H's. We just mentioned that nonpolar things can form these van der Waals interactions with other nonpolar things. And so because that's the case, because the nonpolar portions of the ethanol can form these attractions with these fatty acids that you find on this lipid, that's what ends up potentially pulling apart this fat molecule, causing it to dissolve. So if we were to draw this in a little bit different way, we're saying that ethanol molecules can form van der Waals interactions with fatty acids. If there's enough ethanol molecules around, that could end up pulling apart, breaking apart that fat molecule, causing it to dissolve. Again, so that old adage, like dissolves like, that works, and you can say that in most cases. And for the case of dissolving lipids, this is the reason why that is the case. This is the reason why things like ethanol can dissolve things like lipids. So to write this all down in summary, again, it turns out that nonpolar solvents, for example alcohols, can dissolve nonpolar solutes, for example fats. And the reason why that is because it's due to these van der Waals interactions that can occur between the solvent and the solute. Right? These temporary attractions can form between nonpolar things. To wrap this up, we'll explain one last thing. So at least when I first learned about this, here's what I wondered. So we just mentioned that nonpolar things like fatty acids, even though the electrons are being shared, they can form these temporary partial charges. So temporary plus charges, and then temporary partial negative charges and temporary partial positive charges. You know that water also has partial charges. So if, say that you were to put this fat molecule in a beaker of water, right? The portion of the water that has the hydrogen has a partial positive charge. The portion of the water that has the oxygen has a partial negative charge. So the thing that I wondered when I first learned about this is, well, there seems to be opposite charges here, right? You have a partial positive charge here, a partial negative charge here, albeit a temporary partial negative charge. Wouldn't you expect an attraction to form between these things because they're opposite charges? Likewise, wouldn't you expect an attraction to form between these things because they're also opposite charges? Normally you would, but it turns out that the hydrogen bond that forms between water molecules is stronger than any bond that might form between this and that temporary partial charge. So another way of maybe thinking about it is that if a water molecule has a choice between forming an attraction to another water molecule or forming an attraction to something that only has a temporary partial charge, the preference for the water molecule would be to form its relatively stronger hydrogen bond with another water molecule. So that's why water doesn't really form bonds with these nonpolar things, even though these nonpolar things do have partial charges. It's because they're temporary that water would prefer to form that attraction to something that is permanent, a permanent partial charge. So the hydrogen bond is stronger than any bond that might form between the water and the fatty acid. So kind of a cool thing. Lipid solubility, it doesn't dissolve in water. Lipids don't, but there are things that can dissolve them. You might be wondering, well, that's all well and good. This is a good story. I now know how to dissolve lipids, but what does that have to do with cell biology? Well, in a couple of weeks, we're going to talk about, well, how about if we wrap this up by summing it up in words? So van der Waals interactions are these temporary bonds. What's the reason why water molecules don't bind to those temporary bonds? So it's because of this. Hydrogen bonds between water molecules are stronger than any bond that would form with the temporary charge on a fatty acid. So again, water molecules would prefer to form that hydrogen bond with other water molecules as opposed to the relatively weaker bond that it might form with a fatty acid. Here's how this all relates to cell biology. So again, therefore, water molecules remain bonded to each other and not to the fatty acid. In a couple of weeks, we're going to take a peek at how molecules might dissolve through the membranes of cells. And so it's a kind of an important thing that molecules can move, that some molecules can move in and out of cells, because that's one of the ways that cells can acquire things that it absolutely needs. One of the ways that you can figure out which molecules can move in and out of a cell's membrane is by knowing how lipid soluble it is. So we'll learn that the membrane of cells is made mostly of lipids. So that's what these circles with squigglies represent. And actually we talked about this in the knowledge check assignment. These are things called phospholipids. And so if you have a molecule whose chemical property makes it so that it dissolves in these lipids easily, that would make it easier for it to move through that membrane and into the cell. And so again, knowing something about how soluble a molecule is will tell you about how useful it might be to a cell, or how easily a cell might take it in. And so again, we'll mention more about that in a couple of weeks when we take a look at cell communication.